To be the recipient of the Peter Wildy Prize for the Microbiology Society is an unbelievable feeling, particularly because I've been a member of the Society for many, many years, and I've also heard absolutely awe-inspiring previous recipients, so now to be included in that is amazing. But I think what's important to say here is it really takes kind of a village to do public engagement effectively, so this is very much a team prize, and I would not be here without team members kind of past and present, but actually it's all the other experts and expertise that we get from the other people that do public engagement as well, and just the other fantastic, say, artists and other collaborators that we've been lucky enough to work with over the years. So it's very much a team prize. So the work that led up to winning this prize is, I guess, around our series of kind of, I guess, public events and communication that we do around the gut microbiome. So these are the microbes that reside inside us and the fact that they're really important for our health. So we've kind of had a theme or a topic around guardians of the gut, and there's been a couple of different things that we've done around that area. And I guess this prize is a recognition of the way that we're able to kind of communicate our science and our guardians of the gut to diverse audiences, kind of the public and also school children as well. I think engaging the public with kind of the science that we do is fundamentally important because we want them to come along with the journey that we're also doing as well. We want to inspire the next generation. So I want lots of budding microbiologists coming up and wanting to do this as a subject area. And also I think it's really important to kind of demystify what we're doing as well, because we can bring that trust in as well. And that might actually help people make more informed decisions because they feel more confident because they've got that background knowledge. So I think that's why it's really important to do this type of engagement. So in terms of difficult things that we have to think about, particularly for, I guess, microbiome or microbiology research, is they're invisible. So you can't even see it. So trying to explain to somebody that what you work with, you can't see with the naked eye, that you need a super duper microscope to be able to do that, that's really challenging. And I think the other thing that's really challenging is because we work with microbiomes or microbial communities, we're not working with one individual tiny bacterium. We're working with trillions and trillions of microbes. And again, getting across that sheer scale is also very challenging because obviously that makes it really complicated because each of these microbes does something slightly different for our overall well-being. So it's been able to kind of, I guess, not oversimplify, but to get it down to a level, and again, that's different for different audiences, to get enough information across without people being like, I can't get my head around this, I'm not interested. I think that is the big challenge for microbiology research. I guess some of the ways that we've overcome those issues is making these microbes come to life so they're not invisible anymore. And that's kind of where the theme Guardians of the Gut has come from. So instead of thinking about trillions and trillions of microbes, we've kind of condensed it down into a couple of manageable, key, beneficial microbes and what they do for us in terms of their jobs, in inverted commas, like in the gut as well. And the best way for us to do that was from a visual perspective. So we actually built a giant walk-through gut that was filled with 5,000 LED lights, and the colours of the lights depicted these different microbes, and then people would understand, well, who's there under particular conditions. So for example, if you're maybe being breastfed or if you were formula fed, how that change of a diet can actually influence what microbes are as well. So it's actually physically immersing people in that environment and then understanding that the changing of lights then also meant that there was a change in microbes and what might that mean both short term and longer term as well. And I think that kind of really hands-on activity worked incredibly well from a communication perspective, and people just kind of, I guess, got it because they were in it. We've had this kind of freely available Guardians of the Gut classroom pack, which actually we were lucky enough that the Microbiology Society played a huge role in terms of our grant to be able to kind of make that freely available. At the moment, we're actually working on adding on an additional lesson to that. So getting more involved in outreach, it can be quite daunting if you haven't done it before. So I think one thing that would be good to do would be maybe to see people that are maybe have successfully done that kind of in your local area. So that might be in the department or even at the university level. Maybe going, if there's a public engagement team there, going having a chat to them as well. Because I think you can learn from so much from these experts or people who've already done it, which might be to maybe stop some of the mistakes or redo those same mistakes again. And it gives you a little bit more of a framework about what might work and what might not work as well. And I think that's really important. There's also some other fantastic free online activities that are already out there, which you can probably search for as well. And I think if you just got a passion for it, you'll probably be able to find something or find somebody. And it might be that it's not necessarily you do it as part of your team. There might be other activities you can do outside, or big central activities, big science festivals, for example, where they want volunteers. And then you can dip your toe and see if you like it or not, and think about how you could move forward as well. So in terms of thinking about our Guardians of Gut, our physical exhibit, we're kind of thinking at the moment about how we can maybe kind of reboot that or rebuild that, but in such a way that it actually becomes a little bit more portable. So we can maybe share it around a little bit more as well. That would be really exciting. And also been working with colleagues actually at the University of Birmingham at the moment on a kind of social science project, actually looking at antimicrobial resistance and actually bringing art into that theme, and how we can kind of put different maybe objects of art and kind of relate that back to microbiomes or antimicrobial resistance to really get people thinking about things as well. So I guess that's kind of short term. I've got other huge plans, longer term, but we're going to have to see if we get there, and importantly, if we get the funding to get there as well.
Thank you very much for that really fantastic introduction, Doug. And just to confirm that the prize was decided last year before Doug was president, so before anybody thinks that there's nepotism involved. So let's get started. So first of all is a huge thank you to the Microbiology Society for awarding me this very prestigious prize. I've been coming to the Microbiology Society annual meetings for many, many years, and they're really a highlight for me, and mostly because I get to catch up with fantastic friends and colleagues, but I also get to hear about the amazing cutting-edge science that's happening. And I think it's really important for me to keep up to date with this science, because then that potentially feeds in also to the communication that we do both personally and as a team as well. And the other thing I'd like to say here, that this is a personal prize, but of course this is very much a team prize, and you're going to see how many people have been absolutely critical for all of our public engagement and science communication work throughout my career, both kind of right at the start and kind of where we are now as well. And I think the other thing just to say is that, coming to these prize lectures is always like amazing. And I think for me, particularly for this prize, some of the other previous recipients and then also being kind of counted in their numbers is a huge, huge honor. Particularly when Aruka got hers in 2023, she was absolutely outstanding. And I hope I do even a small amount good job as she did as well. So let's get started. So first of all, I want to introduce who the prize is named after, because when I started doing a bit of digging into his career, it was weirdly similar to mine in terms of like where we've kind of been on a journey. So Peter was a virologist, I'm more of a bacteriologist, but he did training in Cambridge. He then was at the University of Glasgow, actually where I did my undergraduate degree, and obviously Cambridge was PhD at the Sanger as well. And I'm currently now chair of microbiome research at the University of Birmingham, where Peter was as well. And also slightly freakily, he was also president of then the Society for General Microbiology, or Microsoc, roughly around when I was born as well. So there's kind of lots of I guess, similarities there. But it was really his commitment to science and science communication and education that kind of this is what the prize was named after. And I had a quick look to see if there was any fun quotes or anything that I could attribute to him that might kind of semi-segue into the presentation. And there's one here from the Lewis Carroll poem, Hunting of the Snark, which hopefully as we go through the presentation you'll get a bit of an idea about, because when I read it, and this was a quote that he gave in 1962 in a symposium about the structure of viruses, but actually it sounds very much like how we built our giant walk-through gut. So we'll get to that a little bit later. And then the next thing I want to do is just thank those that nominated me, so fantastic colleagues James, Alan, Leslie and Willem. And I just want to highlight here as well, as it's so important for colleagues to nominate each other for prizes. So do think about doing that. And in fact, the prize panels that actually decide who gets the prizes are open at the moment for the Microbiology Society, so please do sign up for those as well. And I think the 15th of June is the deadline for the 2026-2027 round, so please do think about that. Next, perfect, is it takes a village to do public engagement, and these are just some of the fantastic people that I have had the privilege to have in my teams across the year and who have been really important for huge numbers of engagement that we do as well. And really without them, we wouldn't be able to do particularly some of the bigger engagement activities and have their enthusiasm really shining through as well. And they're also really supportive if I have to go and do any kind of media work as well. They're incredibly supportive and kind of help behind the scenes to facilitate that as well. So again, very much this is a team prize. And just one particular individual that I would like to highlight who will pop up throughout the presentation is Dr. Jenny Rant. Unfortunately, actually, Jenny's not here. Jenny's now an incredibly good mate. This is us on a hike together. But she's a public engagement expert and consultant, and she's really helped shape our public engagement activities and really take them to the next level. We all talk about having expertise in different areas of science, and actually then engaging with those experts from a communication perspective is so important. We have networks, we build networks at the Microbiology Society annual meeting. Jenny has networks, she knows artists. So it's like these people that we would not necessarily come into contact with, but we can potentially facilitate that through each other's networks. And actually I started working with Jenny kind of by accident, but we'll come back to that a wee bit later in the presentation. But I just want to highlight her because she's been absolutely pivotal for everything we have done. She makes everything just better and brighter. So a question for the audience: what's the biggest challenge you face when explaining your research to non-scientists? And I think one particular thing that we have as microbiologists or working in the microbiome field is that microbes are invisible. So it's really hard for people to kind of relate to them because we can't see them. The other thing is the microbiome, in terms of these microbial communities, they're incredibly complex. There's trillions and trillions of microbes. How do we explain that to the public? And how, particularly because each of these microbes have a slightly different function as well. Often a misunderstood field. There's a lot of hype in the area. So in terms of thinking about, probiotics or gut health, but the public might not understand what the nuance is behind that as well. And that's a real challenge to communicate that. And of course, for us as scientists, it's this oversimplification plus also technical overload. So it's just finding that right balance, which can be really, really difficult and normally is an iterative process as well. Science can often feel intimidating. People just kind of maybe don't get it, which is why I think it's really important to be effective communicators as well. And having science accessible is also very empowering, I think, and for people making particular decisions as well. And I think from our perspective, the microbiome affects everybody, so it's really important that we're part of that conversation as well. So this is kind of where we've come in, which is like, why would somebody not think about the microbiome? And I'll give a couple of stories and case studies about how we've done that across the years. So my journey into public engagement, as Professor Gordon Dugan's already mentioned, I did quite a lot of public engagement kind of in my PhD at the Sanger Institute at the University of Cambridge, kind of working with school children. And that was really fantastic to be able to do that and kind of dip my toe into that area. But really, actually, it was when I moved to go over to the Irish Sea, over to the APC Microbiome Institute in Cork in Ireland, that I really got to see how public engagement is done at scale and done really, really well. So a big shout out, and there's a couple of colleagues from APC that are here as well, and I know that they're still very active in public engagement. But again, there was people that were leading on public engagement and then seeing about not just reaching one school, but actually reaching schools across Cork and actually County Cork. And this was done at the institute level, was absolutely amazing. And I remember one particular time where I went into a primary school and the teacher came up to me after and she said, some of these kids literally don't engage at all, and I have never seen them so excited in my whole entire life. So it was really kind of amazing to understand that. So when I kind of moved back to the UK and started at the University of East Anglia, I was looking for other public engagement activities, but it was quite hard to know what to do. So I got pulled into some kind of individual ones where I was kind of giving public talks at a number of different science festivals like Cheltenham and Edinburgh, for example. And then also completely weirdly, I did something very, very early in my career, which was BBC Horizon, which was one of the most intense, crazy experiences of my life, because of course it's like filming, but it really gave me an insight into how engaged the public was in terms of the microbiome as well, because I actually dealt with the kind of the families that were on this. It was actually kind of thinking about allergies and the link to the microbiome and getting so many questions from these families, like one of them saying, well, my son has really bad allergies, my daughter doesn't, why might that be? And kind of that thirst for knowledge really kind of drove me into figuring out what might be important. So I was really kind of keeping an eye out for kind of what we could do with our science. And what is our science? So we work on microbiomes, but we predominantly work in understanding how these microbial communities are really important in the early life developmental window. So this is these are really our microbial starter packs that are transferred vertically during childbirth, and I'll talk a little bit more about that. And they're fundamentally important for our overall health and well-being. So this is because these microbial colonization basically overlaps with the early life developmental window. So it goes hand in hand with all these different physiological processes that are getting started in us as hosts, and our microbes are colonizing around that time. And they're really important for numerous different aspects of overall health, so infection resistant, and a quick shout out to Raymond's got his talk later today, so go and see it. Luke has got a poster today, so go and see that as well. So you can actually find out about the science that's underpinning some of the communication we're doing. The digestion of food, and I'll talk a little bit about that in terms of milk-based diet in early life. Immune programming, and Charlie's got a fantastic poster about how specific microbes are really important for programming the immune system as well, so please go and see that. Really important for keeping our gut barrier tight, because there's many different conditions that are linked to a leaky gut barrier. Metabolism, Doug's already mentioned the gut-brain axis as well in terms of brain or cognitive development and kind of well-being and resilience, and a whole lot more, but we'll come on to that in a moment. So one of the microbes, we like to work on lots and lots of different microbes, but I guess one of our kind of superheroes that we work with is Bifidobacterium, and some of you may have been in this, the microbiome session where I gave a presentation yesterday afternoon. But just to highlight here that for those of you that are bacteriologists, you work on ones that are rods or cocci. Ours are Y-shaped, so they're beautiful and fantastic. And this is just an electron micrograph here of Bifidobacterium, and we'll come back to how that actually inspired a bit of art a little bit later. But actually, Bifidobacterium has been known for many, many years, and actually this is just a hand-drawn diagram by Henry Teaser at the Institut Pasteur in 1899, where he was looking at a group of babies that had pediatric diarrhea, and he had a group of healthy babies. And when he looked at the healthy babies, he noticed that they had these Y-shaped microbes that were absent in the babies that had the diarrhea. And then he was able to take these Y-shaped bacteria, put them into the kids with pediatric diarrhea, and then they didn't have diarrhea anymore. So we have known, and this is the other, I guess, overhype. Microbiome's not a new field, probiotics are not new, but it's really important about kind of managing those expectations for the public as well. And the other thing that's very important for us is that Bifidobacterium is one of these microbial pioneers. It's one of these first colonizers in the first hours, weeks, and months post-birth. So this is just our Bifidobacterium here, being a pioneer. Welcome to the gut, or welcome to the jungle, I guess. And it's just important to understand that this microbial ecological succession and these initial pioneers are absolutely fundamentally important for setting up health both short and longer term as well. So what shapes our microbiome? And I'm not going to talk about all of these different factors, but these are microbial communities, and we have to treat them like ecosystems. And actually, that's one of the concepts we try to get across when we talk about microbiomes to the public. And because, particularly in early life, these communities are just getting up and running, they're very susceptible to perturbations or disturbances. And some of the major factors that might change or shape our microbes are birth mode. So if you're born by cesarean section, or vaginal delivery. So kind of vaginal delivery, you get this vertical transmission of beneficial bacteria from mum to baby. And this can be interrupted via cesarean section. And one of the main microbes transferred vertically is actually Bifidobacterium. Diet. So in this case, obviously, the milk-based diet that's obviously kind of prior to weaning or the introduction of solid food, and that might be breast milk, formula milk, or a mixture of both. And we're going to come back a little bit to that about the importance of Bifidobacterium in that dietary environment as well. And the other thing that's kind of already been, which we've already heard many talks about, and we will continue to do over the course of the annual meeting, is antibiotics. So antibiotics, say a medical marvel, but they don't discriminate between potentially the bad and the good guys. And Bifidobacterium, for example, is very susceptible to antibiotics as well. And of course, the other thing about antibiotics, not just killing beneficial bacteria, there may also be consequences for antimicrobial resistance as well. So just to say that I want to, and I won't go into too much detail, but I just wanted to highlight that I think the whole way through that we've been communicating as a team about our research, or if I go on to like do any media stuff or I write about everything, it's really underpinned by the fundamental science that we do. So we do work very much in the early life window, and we understand ecological interactions, we do clinical studies, we do some preclinical studies as well, and we're really thinking about kind of harnessing this knowledge for the betterment of human health. But I think it really makes it easier, particularly I think as a team, when you're able to communicate your science, like as the first step into public engagement, because you get it, because you're working on it. And then as you gain in that confidence, you can start to maybe think about more broader kind of topics that you might feel comfortable talking about as well. So what are the consequences of microbiome perturbations? So what happens if we disturb these burgeoning microbial communities in early life? Well, here's the kicker. So basically, every single type of disorder that you can think of, particularly from a human context, has been linked to changes in the microbiome. So you can very much understand why there's a complete fixation by the public on understanding microbiome science, and also thinking that we're going to cure everything, in the next 5 years, which would be amazing. And actually, there's quite a lot of amazing things happening, and there will be some fantastic microbiome-based therapies that are already on and coming onto the market. But we've still got a long way to go, so it's really important to kind of, I guess, meet those expectations. And the other thing, and I don't need to say this for this audience, but the other thing that's really important to say that not a load of these studies are association. So you look in a disease population and they've got a different microbiome to a healthy population, but there's so many different confounding factors that might be driving that. So it's also managing the expectation, again, of the general public to be like, there's differences, but what do those differences mean? And I think that's something in terms of why I think public engagement matters for microbiologists. So this is actually a really nice piece of work that actually Leslie and Alan did, I think not last year, just the end of 2023, where actually it's not important just in terms of actually for the public, but actually for the general microbiome community. There's quite a lot of myths that need to be busted out there as well and kind of managing what that is. And I'm also delighted to hear that actually the Microbiology Society, I think as of today, will actually be releasing a guide in terms of thinking about the microbiome and communicating that to the media as well, and thinking about specific facts that are correct and other facts that are not correct as well. So I think that's just really important to say that as well. I think bridging the knowledge gap is really important because microbiology can seem like this complex, invisible type of science, and it kind of helps to bring these microbial worlds to life and hopefully makes it much more accessible for individuals. I think the other thing that's really important for me is inspiring the next generation and also having role models as well in terms of thinking about, well, if they can do it, anybody can do it. And I will testify the fact that I'm standing here giving this presentation. If I'm here, ruddy hell, anybody could do it, okay? Also, I think the other thing that's important to say here is about hopefully it encourages responsible behaviours, and I think that's incredibly important in the current climate as well. So if we can demystify and communicate that science, hopefully we can get people to make those kind of right decisions and think about what they want to do as well, and that responsible behaviour, and that might be hygiene, vaccines, or antibiotics, for example. It's important for science policy and research funding. I think Doug mentioned this yesterday, an amazing initiative that was being led by Tina and Katrin and also the Microbiology Society, knocking out AMR. And I know that communication and education is one of the key pillars that's come out of these workshops that happened in 2024, so do have a look at this as well. And I think it makes us better scientists, and I think that's in terms of the communication that we do. And also that dialogue with the public is really inspirational in thinking about how that might influence our science. And I'll talk about a couple of examples where those conversations have impacted what we do. So what works in public engagement? I think this is something this is all initiative process. You learn as you go along. It's the same as doing science. The experiment doesn't work. You figure out, you troubleshoot it. Why did that not work? How can we move forward with this? And I think it's important to kind of think about all these aspects, particularly thinking about, for example, the first one is, what audience are you going to be delivering this to first? Because, of course, chatting to school kids versus maybe doing a public lecture to maybe older adults in a particular type of setting, it will be quite different. And in terms of particularly the analogies that you might want to potentially use there as well. This two-way communication, so we can obviously just do this by giving them information, but it's really quite fun to have this two-way interaction as well. Clear goals. What do you want? What's your take-home message that you want the public to know? I think that's really important because that helps guide what it is. It's the same as your hypothesis. Let's do this for public engagement as well. Be creative and think about immersive formats, and I'll talk about one of those ones in a moment. I think the other thing is being inclusive and accessible, and I'll talk about one particular thing that really kind of blew my mind that I was involved in down at the Wellcome Collection as well, about how important that inclusivity is as well. evidence over assumption, so actually showing the evidence rather than people coming in with their prior assumptions, or at least challenging those assumptions as well. Leveraging partnerships, we wouldn't have been able to do anything, as I've already said, without the incredible partnerships that we've had as a team, and I think it just makes it into a much bigger, better kind of overall public engagement project. And the other thing just at the very end here, apologies, that's shifted a wee bit, is the one thing I think we forget when we're doing this communication is how do we evaluate it and how do we assess the impact of it? And that's really important because we need to see, have we made a difference? And if we haven't, that's fine, but can we improve things going forward? So I guess one of the things we wanted to do was microbiome to the masses. So one of the first kind of big projects that we were lucky enough to be able to do was Welcome at that time were providing additional funding on top of if anybody had Welcome funding already, and I was lucky enough to have an Investigator Award at that time. So I was really keen to see if we could actually do something really big and bold with additional funding from Welcome for this as well. And the overarching aim, which was a little bit bold, but that's all right, was to increase awareness of the UK public in relation to the gut microbiota. And I thought about doing that in a number of different ways. And this is kind of where Jenny comes in. So I wrote this grant. Welcome said, it's grand, Lindsay, and actually we're really excited about it. But no offense, but you're still relatively early career. You don't actually have a professional background in this. And we're really worried that actually if you take all of this on, you won't be able to do any of it very effectively, which is a fair point. And actually, we think that you need an expert here. So it'd be the same as me having a collaborator for science, for example, that we were doing proteomics, for example. And I'll go and write one of my fantastic colleagues down that would be part of that grant. It's exactly the same for public engagement. So they basically made me go out and find somebody that could be brought into the grant to kind of help us realize this ambition. And that's kind of where Jenny came in. And she's been absolutely amazing. She helps people both in Norwich, kind of across the UK and internationally as well. And one of the things that we did as part of this was to deliver a very specific activity down at the Royal Summer Science exhibition, Guardians of the Gut. This is where the theme comes in. And it was very easy to write this application. Well, not so easy, but it wasn't a massive application. But then we got told we had it, and me and Jenny looked at each other and went, oh, we now need to deliver on it. So this was like, and I'm going to talk about this delivery a little bit in a moment. So I want to just show you Guardians of the Gut as a case study in science communication, and also want to give a big shout out here. This was when I was at the Quadram Institute as a group leader, and I'm still affiliated there, and Andy Chappell's one of the comms team there, and Andy's been fantastic both for this project but many other projects across the years as well. And these are some of our microbes that we introduced down at the Royal Society, and I'll show you about how we made them visible to the general public in a moment. So the making of the gut. So again, this was Jenny and all of her contacts. So this was like a giant walk-through immersive gut experience. And funnily enough, I don't necessarily know the people that would be able to facilitate that build, but Jenny did. So we were lucky enough to have Tim from Tin House, who is actually kind of a set designer for the theatre, which kind of makes a lot of sense now. And we were also lucky enough to also work with the Norwich Hackspace in terms of actually kind of doing all the electronics and helping with the build and the design and thinking how we can make this as good as it can be from a public engagement perspective as well. So we obviously took our inspiration from pictures of the gut in terms of her actually designing the overall build, so diagrams, and then coming up with kind of scale comedy models. And actually, it was a very organic and homemade gut, which was made of basically plastic water pipes, mesh, and a lot of LED lights as well. We had these microcontrollers that were basically the brains behind the gut, so it was amazing because it was actually technically the gut-brain axis, but in an electronic form. And we had 5,000 LED lights that basically were all over the interior of the gut. And we had the 3 different colors, and then the guys in the hack space were able to work with us and were able to program the lights. And they went back to game stations and able to program them for different colors, which then went back to different microbes in particular, and I'll show you a picture about that just in a moment. We then did lots of materials to kind of cover up those lights as well and kind of paint it up to look like a gut. And we also did a cross-section of a gut that kind of was put on the front and rear end of the gut as well. And something I would like to highlight here is this looks like this was slick, easy, professional. We were still painting this at 10 p.m. the night before we needed to go down to London. And we basically then had to dismantle it and then put it on the van and drive down to central London and then rebuild it all again. It was a huge amount of work, but everyone pulled together amazingly as a team. So, and then this is me just diving out the gut as well. But I just want to say about how we made these microbes visible. So the first thing that we did, we actually took our participants on a tour, so those that came to the Science Summer Exhibition. And the first thing we did, which was kind of this welcome to the microbiome quiz. So it was the first one to kind of gauge what people's knowledge were before they even went into the gut. So the first one was like, you know, how many microbes versus human cells do you think that you have? If you took all these microbes, how much would they weigh? If you took all these microbes and you put them in a line, how far would they go around the Earth? Like 2.5 times. And this was a really nice way of kind of getting people to understand kind of how small, but how numerous and how many there were, particularly because we completely mashed up the overall complexity. We had 4 different game stations that basically talked about how different factors might shape your microbiome. And I'll show a little bit about them just on the next slide as well. So birth mode, diet, both kind of both in early life and later life as well, and a course of antibiotics, what that might do. And then these were some of our main players here as well. So each of these microbes, everybody got a wee card to find out what they do. And each of these different we had lights that changed colour for the different stations, and this is chosen one of the postdocs in the team, and you can just see, it's pretty big inside actually, but you can kind of see those different colours as well. And I'm hoping that, well, I'm not hoping, the next one, I just want to quickly show a video, don't worry, it's not too long, but I think it's hard to understand what it was like to walk through the gut without having a wee video, so let's hope this works. Excellent.
So first of all, let's move through the villi.
I hate hearing myself.
So, welcome. As you can see, it's a little bit dark in here, but what we're going to do is we've got interactive game stations that link back to how different factors influence the microbes that we have within the gut. So the first one is actually how you're born impacts what microbes you have. So if you're natural birth, you can see that we've got a number of different colours here, predominantly purple and green, which links back to bifidobacteria and lactobacillus, and those are the types of bacteria you get if you're born naturally. If you're a C-section baby, we've again still got some lactobacillus and bifidobacteria, but we've also got different types of bacteria. And this is because when you're born by cesarean section, the first types of microbes you come into contact with are actually skin microbes.
And
so
hospital
that's
environmental.
What's next? Well, obviously, babies are on a milk-based diet before they move on to solid food. So let's look to see if bottle feeding or breastfeeding, what that does to your microbe. So if you're on formula, you can see that lots of different lights have lit up, which links back to lots of different types of bacteria that you have within the gut of babies that are bottle fed. But if you're a breastfed baby, let's have a look at the change of colours. So hopefully you can see that we've got lots of purple, which links back to BIF, which again is our favourite bacteria. And why have we got lots of purple in breastfed? That's because bifidobacteria are actually able to digest components of breast milk that basically enables it to outcompete other types of bacteria.
And I'll end it there rather than having to put you through the whole thing. But hopefully from that you can see that this is kind of a really effective way of communicating quite complex science in a literally interactive, immersive way, because people had to physically walk through the gut. So just a couple of just photographs from the day. That was us just after we'd finished the build. It was incredibly busy. It was a fantastic week. I know there's quite a few other colleagues out here that have also done their own science at the Summer Science Expedition as well. We were up on the stage as well, which was really nice. So people kind of saw us and then were like, ooh, what's that very homemade thing? And wanted to come over and have a look at it, particularly in comparison to some of the other very slick public engagement presentations from some of the other universities that were slightly better funded than us. So really enjoyed kind of taking our tour groups through the gut as well, almost doing a safari. Got to meet Brian Cox. He kept saying about, oh, well, the solar system and the galaxy is really complicated. And I was like, mate, it's like there's more microbes in the gut than there are stars in the sky. And he was like, really? And I was like, and he was like, big day? I was like, yes, bread and butter, it's fine. So that was quite fun to meet him. VIP that came down, mum and dad, who are actually in the audience at in the front there as well. And we really had a fantastic time. And of course, down at the Royal Society, we also got to do our evening thing where all the team got to dress up and kind of actually take round VIPs and eminent scientists with a glass of fizz as well, which was fantastic. But the other thing that I mentioned before here is it's really important to capture feedback. So we wanted to see if actually, our ridiculously elaborate walk-through gut, if actually it was effective or not. So we got lots of kind of different feedback in a number of different ways, and actually a really easy way to do that as well was just kind of post-it notes. Just a couple of different ones here as well. But a couple of ones I just wanted to highlight is the fact that, yeah, that was brilliant, fantastic Joanna 6, which is fantastic. And I think I probably still spell as well as Joanna does, but that's okay. Best exhibit, and I think it was just really nice to have that feedback. You obviously get verbal feedback, but it's important to get this written feedback as well. And the other thing about what do we want to get across the public, what was our top level aim that we wanted to do? And then I got a post note from one of the school groups that came through and I was like, learning objective, tick. Literally got that completely right, which was like amazing to kind of see that as well. The other thing we were able to look at was a huge jump in website traffic, both to the Institute website and our own website as well. That's kind of an easy way to gauge if there's kind of interest. And we were the second most popular exhibit. We weren't first, but that was because the NASA crew, who had a £70,000 video, won. So I think we did pretty well, actually. So I'll take second. What were the challenges and lessons learned? I think the storytelling, a little bit of humour. We certainly said when we took the kids through the gut that we would, and I kept saying, well, let's get pooped out the other end, which of course they absolutely loved. I've seen most of the adults did as well. Great learning experience for the team as well. I think there was a couple of individuals that were really worried about kind of communicating their science, actually even in terms of science audiences, but in terms of the additional confidence they got from kind of going down and kind of being literally thrown at the deep end and having to speak to huge different audiences was kind of really empowering, I think, as well. But I want to highlight a couple of different things. There was quite unexpected costs. This is Shab making us a fantastic meal in one of the houses. So I hadn't really appreciated. We were all going down to London. We were in Norwich at that time. 10 grand people for accommodation for the whole team. And that wasn't something I'd factored in because I didn't think about accommodation. So that was something that we had to scramble to get money for, but it was really great kind of being in different houses and kind of all sharing stuff together. The talent of the team, so I found out a load of them were incredibly creative and almost like probably would have been artists if they weren't scientists. And actually I think probably we are all artists as scientists anyway. Pink bath mats aren't that common. So I thought pink bath mats would be quite good for the villi, but actually we ran out of pink bath mats and we were like literally driving over the whole of Norfolk to try and find more of them. There's not that many. The other thing, it was July, it was very warm. We got to the point where it was basically physiologically relevant temperature in the gut, like 37 degrees. So we had to get fans put in as well. So it was, yeah, that was a little bit challenging as well. And that's me after we do the build on the first day. It was one of the most exhausting, stressful experiences. The Royal Society people were like, did you not practice like putting it up and putting it down and putting it up again? And I was like, lads, we did this at 10 p.m. last night. You know, and we've literally driven down from Norwich, and you've given us no extra money for this. So it was really exhausting, but absolutely rewarding. And I think one of the things that, again, kind of going back to that thing I said about Brian Cox, that I quite enjoy saying this, is about there's more microbial species on Earth than there are in the galaxy, well, as we know at the moment. And we've still got lots of other microbes to discover as well. It's kind of just a nice way of understanding that. So as part of the summer science exhibition, but as kind of other engagement activities, what we did with the gut, we've also got kind of a comic that went with that as well that we were able to give away to kids in terms of just kind of reading and learning a little bit more about their microbes as they left. And we also have taken the gut on tour to different parts of the UK, including even a music festival, so Latitude Music Festival. So we even camped in the artist enclosure, which was like, it sounds incredibly glam. The only thing was they had showers, and that was it. But that was still quite nice over 4 days as well. And again, fantastic team that went down to do it. So it was amazing listening to some fantastic music and then also doing the outreach during the day as well. And the other thing I did say I was going to mention was about how the public engagement has actually shaped our science in terms of those two-way dialogue conversations. So certainly at the start, I got a lot of questions around kind of the diet, probiotics and inflammatory bowel diseases, including Crohn's disease and ulcerative colitis. So that really definitely shaped some of the work that we're doing. And in fact, that's some of the work that Charlie's poster, so do go and have a look about that as well. Microbiome and cancer, it's like a really hot topic at the moment, but that was something that kept constantly getting brought up by the public, and that's very much stemmed some of the other work that we're doing with fantastic colleagues at the Quadram, including Stephen Robinson's team, and understanding how different microbes might drive anti-cancer responses as well. I think we've already heard this, the gut-brain buzz. Everybody asks about this, and I completely understand why. And now with the move to Birmingham, I'm now speaking to colleagues there in terms of thinking about how we can do some really excellent science in that area, pairing up with excellent psychologists and psychiatrists and trying to bring microbiome to the party as well. And I think it's really kind of sparked a citizen science project as well. So we're currently chatting to the Women's Hospital in Birmingham and thinking about doing kind of a citizen science project looking at pregnant women and thinking about sample collection as well. I've already done some patient public engagement and involvement, and thinking about sending them home packs to collect their own microbiome samples and then send in so we can understand how microbes might be influencing pregnancy and preterm birth. And this has definitely also been sparked by a wonderful colleague, Sarah, at the University of Antwerp. I just want to highlight this if you don't know about her work. She's done an incredible, incredible citizen science project looking at the vaginal microbiome that's really spun now projects all over the world as well. And she is really a leading light in this area. So if you haven't seen this project, please go and have a look at her website. And she did this also beautiful science Nature Microbiology paper that was published a couple of years ago as well. So it can be done to underpin excellent science as well. So beyond exhibits or extending public engagement. So the Guardians of the Gut was great, but it wasn't particularly portable. So it did mean that we couldn't necessarily get out to everybody. And actually that was one of the things that we decided to do next, was kind of how could we extend our reach. And this is where kind of Guardians of the Gut, this free online classroom pack came about that was actually supported by a Microbiology Society award that I got back in 2018 that enabled us to kind of take the concept of a giant walk-through gut and actually bring that into the classroom. And this was very clear because many primary school teachers don't necessarily have a STEM background and they don't feel confident teaching science. And this was one way to potentially give them something that would be easy to do. All the materials are sustainable, so they can be used again and again, and that's really important, thinking also about making sure that this is equitable for all schools as well. And also, if the teachers don't feel confident giving the lessons, there's really bad videos of me giving the lessons, so they can just play those instead. So there's been over 300 local authorities that have downloaded this, because when you sign up for this, we ask, who are you, what are you doing? So that's been amazing to see that as well. And in fact, there's a couple of kids that have contacted me recently asking me about doing an interview for their project, which has been really fun as well. And we've also keep getting fantastic feedback from this, including Jenny actually sat down with some teachers and they were like, have you seen this resource? It's amazing. And Jenny was like, yeah, no, we did it. So it's quite nice having that feedback a wee bit later as well. And actually at the moment, we're currently rebooting this with a colleague over in Canada, Megan Azad. Some of you may know, she's an incredible microbiome superstar researcher, and she's really focused on breastfeeding and how milk shapes the microbiome. And at the moment, we're just working on an additional lesson plan that we're going to add to the 3 that we already have. So the other thing that we've done as part of this is also evaluating. So I mentioned about it's really important to understand impact. Well, we were lucky enough to work actually with a child psychologist at the University of East Anglia, Georgia, who helped us come up with a pre and post quiz. So we could basically determine what was the background knowledge of the kids before they do these lessons and how does that improve after we've done them as well. And just wanted to highlight here that even though the children might have a different kind of, I guess, level of understanding at the start, after they've done the lessons, they all come up to the same to show how effective that is as well. And we also do a quiz 6 months later to find out if they still actually remember anything, rather than just immediate, and they normally do, which is absolutely fantastic. Other things that we've done as well is microbiome in the media. I think Dougal already mentioned this, but it's utterly terrifying doing TV or radio. But if we don't do it, someone else is going to do it, and you're going to shout at the TV saying, why are they doing it? So maybe think about doing it yourself as well. I think the first thing I did was after doing Horizon, I went on to BBC Breakfast. It's live TV. And there was one question that got asked right at the end of the interview, which is would you eat toast off the floor? And I thought I'd done this really good dramatic pause, pondered and thought about it. And I was like, well, yeah, I would actually. But in reality, what happened in real time was I got asked the question, and I just went, yeah. And then they stopped it. So that was semi-traumatic, but anyway, I got over it. And then I got reintroduced for the next segment of the interview going, oh, this is Lindsay Hall, who'd eat toast off the floor. So anyway, still do stuff for the newspapers as well, crazy headlines like the Daily Mail, but you can try, and actually be fair, most journalists will actually sit and listen to what the science, you're like, that's wrong, please think about doing it in a different way, but they still need to have something that's a bit engaging. We've done numerous different podcasts, we've done numerous different TV shows as well, like Save Money, Good Health, and then relatively recently, I was also the reboot of Gillian McKeith in You Are What You Eat, and in fact, I'm even on IMDb. I need to put my photo on, which is very exciting. And I play self, so it's not particularly exciting. But yeah, so I did the iconic poo test, so I was that person. But I'm also Scottish, but actually I've got a proper PhD, as Doug will testify to, rather than what Gillian McKeith has. Other things I've done is also radio as well, and just one thing here that most recently, just end of last year, I did something down in London, went down to Greenwich to a comedy club, comedy people, terrifying, but it was really, really, really a lot of fun, and hopefully this little clip will just play now.
Now, Professor Lindsay Hall is a microbiologist and chair of microbiome research at the University of Birmingham. She leads a dynamic team that is on a mission to unravel the mysteries of how the microbes that live inside us keep us healthy. Lindsay, what does your work involve?
Well, my work basically involves poo.
You went to university to study poo. Did you get a number 2-1?
I did, actually.
Yeah. Professor.
So, and I did get a 2-1. And it was great that I didn't know I'd ever get to use that as a joke in a comedy show. But there was just another way of thinking about kind of doing it. And then just last couple of slides, we've also been lucky enough to work with artists in terms of, we also had an artist in residence at the Quadram Institute, Jenny Pedley, who did a number of different work, both with the team and with the public as well. And I did mention I was going to highlight here. The other thing that I was lucky enough to be involved in was I was one of the scientists that was involved in the new Wellcome collection, which is just next to the Wellcome offices just on Euston Road in London. It's their permanent exhibition, well, a 10-year exhibition. And it was thinking about art, but actually making sure the science was like underpinning that. So they already had a lot of artwork that they were able to choose from, but they also wanted to do something really interactive and think about working with those that were visually impaired. And lucky enough to work with artist Tasha Marks to actually create, using an electron micrograph, of a 3D model of bifidobacterium that smells like breast milk. So if you go down to London, go and check it out, it's amazing. And I've even got my name on a plaque, which is like ridiculous, but really, really cool. And again, thinking about how we can do that more effectively. I think the other thing that's important to say is we can talk to the public, but the other thing that's really important from a science communication perspective is actual real-world impact in terms of policy and also clinical work as well. So this is just kind of the website that you can go to that searches the House of Commons or House of Lords and looks for specific key terms, and this is me just typing in microbiome. And actually, if I'd done this probably last year, there would be hardly any notifications, but there's been much more recently because, let me get this right, Natalie Bennett, Baroness of Bennett of Manor Castle in the House of Lords, has kind of been really kind of championing microbiome as well. And I've been lucky enough to go down to the House of Lords actually with a number of colleagues here, and it's actually been led by Professor Leslie Hoyle at Nottingham Trent University, and thinking about kind of bringing microbiome into the agenda for the UK government and just UK science in general as well. I also did the Royal Society pairing scheme last year, which was absolutely fantastic, and learning kind of about kind of, you know, bringing science into policy and what that might mean as well. And this is Gareth, who I was paired with, which was absolutely fantastic. Been able to kind of contribute to a number of different kind of, I guess, white papers as well about the microbiome. The other thing that's really important from a clinical perspective is continuing professional development. So I've been involved by other efforts, maybe led by others as well, in terms of thinking about bringing microbiome to the agenda for clinical doctors and why it's really important to think about the microbiome in the context of what they're doing, particularly, for example, for antibiotics as well. And also I've been lucky enough with some of the work that we've done in terms of our work with probiotics and preterm babies, that we've been able to be, our work and that of others is cited on the World Health Organization clinical care guidelines for preterm babies as well. So it's kind of thinking about science communication in its broadest sense. And then just last two slides. So the challenges here is that, as always, I've kind of been talking about this the whole way through, is time. And I know everyone's really, really busy here. And it's really hard to find the headspace to be able to do that as well and do it properly, but I think it's really, really important to avoid misinformation, which I think, and again, in the current climate is so important. And it's also navigating that misinformation, pseudoscience, and that oversimplification as well. Finding the money and finding the support, and that might be the support, obviously financial support, but also the support like, you know, Jenny and her incredible network that she had that was able to make us realize some of the ideas that we had from our lab work. And again, just I think this is important in terms of the brain microbiome, and everyone keeps asking me about this, and I know many other colleagues in the audience as well, and that's why it is really important to get out and kind of, you know, say about why this oversimplification, or if somebody shows one scientific result, is that actually correct or not? And if it's not correct, why is it not correct? And explaining that. So just to finish off, and I think hopefully I'm on time, why public engagement is worth it? Well, I think microbiome and microbiology research really, it belongs beyond us working in the labs or working on our computers, or like me, beyond me writing emails to try and respond to everybody. I think it's really important to do that as scientists as well, and honestly, it's a lot of fun. It's hard work, but it's a lot of fun. And I'll just leave you with a wee quote from a wee girl that said, you know, when I grow up, I want to be a microbiologist, just like Professor Pooh. And I'll end it there. Thank you very much.